Hello everyone, and welcome to the virtual Biogeochemical Cycles Lab. We're going to be working outside today, but before we do that, I want to review biogeochemical cycles in the Indian River Lagoon. The term biogeochemical cycles refers to the movement of elements or compounds like water throughout different locations in the biosphere. Today we're going to be focused on water, carbon, nitrogen, and phosphorus. However, cycles including other elements and compounds, like sulfur, also occur. We'll be studying these 4 cycles within the Indian River Lagoon. You probably know a little about the lagoon already, but I'll go over a few characteristics just to review. First, the lagoon is a brackish body of water. This means it has both fresh and salt water sources, and the mixing of these 2 types of water produces a moderate salinity. In addition, the lagoon is not a true river as it is influenced by tides. The Indian River Lagoon is extremely biodiverse and includes a variety of different plants and animals. However, it is typically fairly shallow with an average depth of only about 4 feet. The lagoon stretches along 40% of Florida's east coast, and because of this includes both temperate and subtropical ecosystems. As the Indian River Lagoon is a body of water, it plays an obvious role in the water cycle. The plants and animals that live in the lagoon are also involved in biogeochemical cycles as well. Excess carbon dioxide, nitrogen, and phosphorus can have a negative impact on the lagoon, so it's important to study and understand these cycles as well. Like I said, we will be discussing 4 different cycles today, starting with the water cycle. The main components of the water cycle include evaporation, condensation, precipitation, runoff, and storage. Evaporation is mainly affected by wind and heat and causes droplets to be suspended in the air. There is a term for this that you're probably very familiar with, which is humidity. Today, the humidity is approximately 84%. Condensation occurs when water droplets rise, come together, and cool. When many water droplets condense in the same area, they may form clouds, which can then travel using the wind. When clouds become oversaturated with water droplets or too heavy, rainfall and precipitation will occur. Increased rainfall may occur with low pressure systems as it increases the amount of air rising and the number of clouds being produced. The salinity in the Indian River Lagoon may be highly affected by the amount of rainfall. Once the rain reaches the surface of the earth, it will run off to low-lying areas and then be stored in these low-lying areas until it is evaporated again. So essentially, the lagoon acts as a water storage system. It's important to understand that water continues to move while it is held in storage. In the Indian River Lagoon, water is mixed and moved with tides and currents. Moving on to the carbon cycle, the main components are photosynthesis, respiration, decomposition, and combustion. Plants utilize carbon dioxide to convert carbon into energy for growth using photosynthesis. These plants may then be eaten by animals who will respire and release carbon dioxide and also excrete carbon in their waste. Eventually, both plants and animals will die and decompose, releasing carbon back into the environment. There are many plants and animals in the Indian River Lagoon involved in the carbon cycle. Some examples of plants include seagrass, mangroves, palm trees, sea oats, oak hammocks, and more. Some examples of animals include fish, birds, manatees, snails, worms, crustaceans, and many more as well. Combustion is mostly an anthropogenic process caused by the burning of organic materials. This can release excess carbon dioxide in the environment. Bodies of water act as a sponge and soak up this carbon dioxide, but it can cause them to become more acidic. This is monitored by measuring pH, which we will be doing today, so keep in mind that it can be affected by the carbon cycle. The third cycle we'll be discussing today is the nitrogen cycle. The main components of this cycle include fixation, consumption, excretion, nitrate production, and nitrogen gas release. Nitrogen is fixed by bacteria at the base of the food chain. As the food chain continues, nitrogen will be consumed by animals and excreted as ammonia waste. This ammonia is then converted to nitrate by soil bacteria and further converted to nitrogen gas and released to the environment. The last cycle we'll be discussing is the phosphorus cycle. The main components of this cycle include rock exposure, erosion, biotic utilization, and rock incorporation. Over time, rocks will become exposed to elements due to geologic events and eroded. This frees phosphorus to be used by living things, which will eventually excrete it back into the environment. Then phosphorus is available to be formed into rocks once again. We will be measuring both nitrogen and phosphorus during today's activities. Hopefully this will allow you to remember how these cycles work and understand why they're important. Now let's go over some of the materials we will be using for today's activities. Okay, so here are most of the materials that we are going to be using. So first, these are sediment sieves. Basically how these work is each divider has mesh and they're in different sizes. So the biggest particles that are in the soil or the sediment get stuck at the top, and the smallest can make their way all the way to the bottom. So it will separate out the different particles that are in the sediment according to their size. Then we're going to be using a pH meter, which looks like this. This is very easy to use. You simply remove the cap, turn it on, and hold the probe end into the water until it stabilizes on a reading. You just want to make sure that you rinse off the probe with DI water between every different sample. This is a refractometer which measures salinity. How this works is using a pipette, you take a sample of the water, open this little door here, and you put a thin layer of water over the lens. Then you're going to close the door, hold it up to your eye, and look through it so it's pointed at the light. And on the inside, you'll see what basically looks like a ruler with a white section and a blue section. When those two sections intersect, that's the measurement that you want to take for your salinity. Remember, the lagoon typically has a moderate salinity, so we should be expecting to see that when we measure it with our refractometer. Now these are both strips that we're going to be using with the soil. We have a phosphate test strip here and nitrate here. To use these, you hold a test strip into the water for a set amount of time and then bring it out, and there is a color guide along the bottles that you use to compare and give you your reading. This tube will allow us to take a sediment core. Basically, you simply push it into the soil until you reach this line, and this cap helps create a suction effect so you can pull it out and all the soil will be stuck in the bottom here. And that will give us our soil sample, which we can put into the sieves and take our other measurements on. So we'll be going outside. We'll do all of the water measurements outside and some of the soil measurements outside as well. And then we'll also be bringing some soil back into the lab for further measurement. Let's go.
So this is the Indian River Lagoon. This is where we're going to be taking both our water and sediment samples. And right now we are on the north side of the campus, right behind the Johnson House. So let's get started.
Okay, so we have our samples of both water and soil, and we are ready to start testing things. Okay, so nitrate will be first. And now I need to wait 1 minute. So while we're waiting on the nitrate, I will work on the phosphate. So for the phosphate, I need to fill a small tube. And I'll be inserting the strip into the tube. And I need to invert the tube 5 times. Now I'm going to remove the cap of the tube and the test strip. So here's our color chart and here's our water. So I'm seeing it as 100. Down through the water, see the color. So that is the color you want to compare to your colors here. So again, in my opinion, that's 100. If you're seeing that color a little bit differently, that's okay. You can record what you see. And this is for sample number 1. Let's go back to our nitrate and see if we have results for that. Here is our nitrate strip. You can see we really have no color change. If we had nitrate present, we would have seen some degree of pink. So 0 nitrate present in the water for sample 1. So now we are going to repeat those 2 tests with our second sample of water. Okay, let's take a look at our phosphate test results. So again, here's our color key. You want to match the color of that water looking down through the tube to the colors on the bottle. So again, I'm saying that's somewhere between 0 and 100. This might be a little lighter in color. So again, that color is the color you want to identify. Okay, let's take a look at our nitrate results. So again, you can see we don't have any color change here. Again, we're looking for a pink color, so no nitrate here either. All right, we're ready to test our third sample for nitrogen and all right, so again, these are the possible colors for our phosphate test. And you want the color you see looking down through the tube. So again, I'm going to say that's between 0 and 100. Looks very similar to the last one we did, sample number 2. Here's the key one more time. And there's the color. Again, our nitrate test did not show any color change. We're looking for a pink color here, so 0 nitrate. Let's test the pH for our 3 water samples, starting with sample number 1. Alright, this has a pH of 9.43. Here is sample number 2. Let's test the pH. Sample number 2, pH is 9.43. This is sample number 3. So 9.40 for the pH for sample number 3. Okay, so let's test salinity. I have a pipette full with a little bit of water from sample 1. So I'm going to use the refractometer and get a reading. Okay, I'm seeing a pretty high salinity here, about 32 parts per thousand. Okay, we're ready to test. Again, we're seeing high salinity. I'm going to say this one is 31. 31 parts per thousand. All right, this is our last salinity measurement for sample number 3. This one looks to be 32 parts per thousand. Now we are going to separate our first soil sample out into the particle sizes. So I'm going to put the sample on top of our sieves here and separate it out. Okay, here's our first and largest size class. These are the coarse shell. Here are the fine shells, our next size class. This is our coarse sand. Here we have our fine sand. And lastly, our silt, which is the smallest particle. So what you want to do now is compare all of the particle sizes to each other and decide the percentage in each size category. That's what you'll be recording on your data sheet. Now let's look at the particle sizes for sample number 2. Here's our largest size class. These are the coarse shells. Here are the fine shells. Here is the coarse sand. And the fine sand and the silt. So again, come up with a percentage for the amount of material in each size class and record that on your handout. Now let's measure the particle sizes from sample number 3. So here is our first and largest size class. These are the coarse shells. Here's our second size class. These are the fine shells. Here is the third size class. This is the coarse sand. This is the fifth size class. This is the fine sand. And the last size class, this is the silt. Compare all of the amounts in each size class and estimate a percentage of material in each size class. We are back in the lab and ready to test our soil samples. So I have approximately 125 milliliters of our soil in each of the beakers, and I've also added on about 100 milliliters of DI water. For these strips to work, they have to be submerged in water, so it's important to add that in. So what I'm going to do is I'm going to give each of the beakers a stir to try to dissolve as much of the material from the sediment into the water, and then do the test strip. Okay, taking a look at our test strip, we really don't have any color change here. Remember, we're looking for a pink color, and there are no pink shades on our test strip, so no nitrate. And then I will try to give you a closer view of this test as well. But again, we want to look down through the top and then compare it to our blue colors on the side of the bottle here. Okay, so to me this appears a little bit darker than what we were seeing with the water. I'm going to say between 100 and 200, whereas with the water we are seeing between 0 and 100. So again, I'll try to give you a close-up view to show you what I'm seeing as well. So we're going to do the exact same thing with our other two samples. Alright, again, so my view for the phosphate test is between 100 and 200, again a little darker. For the nitrate test, again, no pink shades, no color change here, so that will be 0 nitrate. And again, the same for sample 3. For phosphate, I'm seeing between 100 and 200. And we do not have any pink on our nitrate test strip, so 0 for nitrate. So now we're going to move on to test the amount of salts that are present in the sediment. This is going to be a very rudimentary test, just to kind of give us a bit of an idea or a range for how much salt might be present in our sediment samples. Okay, so like I said, now we are going to try and look at the salt in the sediment. So again, what I'm going to do is stir the sediment around. So hopefully some of the salt that's present in the sediment here will be dissolved in the water when I stir it around, and then we can see some of that salt through the refractometer. So one thing I do want to be clear about, when you're looking at salts dissolved in water, that's typically referred to as salinity. However, salt in sediment is referred to as conductivity. So two different terms. So when the salt is in the sediment itself, it's conductivity. There are meters that can test for the salt directly in the sediment. However, they're quite pricey if you want an accurate one. So I'm going to give you a reading that's pretty general. I will give you a reading of a low level of salt if the salinity is between 0 and 10, medium if it's between 10 and 20, and high if it's 20 or higher. So I'm going to go ahead and get started. Okay, I'm seeing a level between 10 and 20, so we're going to give sample 1 a medium rating for the salinity or the conductivity of the salt in the soil. Same thing with sample number 2. This has a medium salinity. And the salinity for sample number 3 is actually slightly below 10, so we're going to give this a low salinity. We also, of course, want to test our pH. Again, we have our pH meter here, which is super easy to use. Just hold the probe in the water until it stabilizes and then take a reading. Okay, so our pH for our first sample is 9.73. Our pH for our second sample is 9.76. And our pH for our third sample is 9.82. We have now reached the end of today's lab. You should have the data table completely filled out, but if you need to rewind to get some of the data you missed, feel free to do so. One more thing I want to mention, there are actually different types of soil classified according to their characteristics. These soil types include clay, sand, silt, peat, chalk, and loam. You probably know just by looking at it that we were working with a sandy soil today. However, sand is classified according to its larger particle sizes and typically has lower nutrients. Hopefully you enjoyed today's lab and learned more about biogeochemical cycles and the Indian River Lagoon.